In the last lesson, we began to think about how neurons talk to each other through the process of chemical synaptic transmission. An action potential is initiated at the axon initial segment. It travels down the axon, reaches presynaptic specializations where calcium influx occurs, and that drives neurotransmitter release. That in turn acts upon receptors in the postsynaptic membrane, driving postsynaptic potentials. In this lesson, we're going to take a closer look at what happens inside the presynaptic specialization. How can the action potential drive neurotransmitter release on such a fast timescale? There are three key events that need to take place in the presynaptic specialization. First, synaptic vesicles full of neurotransmitter need to dock and be primed to be release ready in immediate apposition to the presynaptic plasma membrane. The vesicle then waits until an action potential invades the terminal. That then drives an increase in calcium concentration, and then in the third step, the increase in calcium drives fusion of the vesicular membrane with the plasma membrane, thus driving exocytosis of the neurotransmitter inside the vesicle. Let's begin by thinking about how calcium rises occur in the presynaptic bouton. The action potential is traveling down the axon. It reaches a swelling where there are many synaptic vesicles present, and in addition to these synaptic vesicles, there's also a high concentration of voltage-gated calcium channels. Voltage-gated calcium channels are similar to voltage-gated sodium and potassium channels, except they have a high permeability for calcium rather than for any other cation. The voltage-gated calcium channels then also have a similar voltage dependence, and so if this is a membrane potential across the plasma membrane in the bouton, and this is the open probability of a calcium channel, then at hyperpolarized potentials the open probability is low, and then as we get more positive than around minus 40 millivolts, the calcium channels begin to open, and they can then let calcium flood into the cell. Remember, there's about 2 millimolar of calcium in the extracellular space, and at resting conditions, there's about 100 nanomolar of calcium, free calcium ions that are present inside the cytosol. So there's a strong gradient for calcium to enter when the open probability of the calcium channel increases at depolarized potentials. So let's see what happens when an action potential invades this presynaptic specialization. Here we see the action potential in a nerve terminal. It has a duration of something like 500 microseconds or 1 millisecond. The upstroke, as we know, is driven by the activation of the voltage-gated sodium conductances. The voltage-gated potassium conductances drive the repolarization. And smaller than those sodium and potassium currents, also a calcium current is found in the presynaptic terminal. It's not activated as rapidly as the extremely fast voltage-gated sodium conductances, so there's some delay before the calcium current turns on. And it turns out that the peak of the calcium current is somewhere on the falling phase of the action potential, so delayed some 100 microseconds or so from the peak and onset of the action potential. This influx of calcium, the calcium current flowing through the voltage-gated calcium channels, then increases the calcium concentration in the immediate vicinity of the synaptic vesicles. And it's that increase in calcium concentration that directly drives vesicle fusion. It turns out that there's an extremely steep dependence upon calcium concentration and neurotransmitter release rates. On this graph, which has logarithmic axes in both the x and y scale, so it's a log plot, you see a very steep dependence upon the cytosolic calcium concentration inside the nerve terminal and the release rate, the number of vesicles being released per millisecond. You'll see that there's a range of calcium concentrations here that goes from 1 to 10 micromolar, so this is a tenfold increase in calcium concentration, whereas on the y-axis you'll see that the release rate increases by a factor of 10,000. you can plot a line through data points, and it turns out that there's a nice linear relationship between calcium concentration and the vesicle release rate, and it has a slope of around 4. That's the so-called Hill coefficient, and that tells us that if there's a 10-fold increase in calcium, then there's a 10 to the power 4-fold increase in release rate. 10 times calcium, 10,000-fold increased release rate. And here's the relationship here then. The release rate goes as a 4th power of the cytosolic calcium concentration in the nerve terminal. So increases in calcium, irrespective of action potential or membrane potential, drive neurotransmitter release. At resting calcium concentrations of around 100 nanomolar, the release rate is very, very small. It's non-negligible. There is still a small amount of vesicles that are fusing spontaneously, even at resting levels. But that release rate increases very dramatically in the presence of calcium. So calcium is really the direct signal that drives vesicle fusion, and it does so in an extremely steep dependence, where small increases in calcium cause huge changes in neurotransmitter release rates. In order for the calcium to be able to work, there must be calcium-sensitive binding proteins that bind the calcium and translate that into the exocytosis of the synaptic vesicle. In addition, for the process of exocytosis to occur on the 100 microsecond timescale, there must be an extremely efficient machinery that's able to couple the calcium signal to vesicle membrane fusion events. Here we see a schematic drawing of a synaptic vesicle that's in immediate apposition to the presynaptic plasma membrane. So these two lines here represent the phospholipid bilayer of the plasma membrane, and the two black lines here around the vesicle are the phospholipid bilayers of the synaptic vesicle. The membrane of the synaptic vesicle is almost identical in terms of its composition to the membrane phospholipids of the plasma membrane. The synaptic vesicle, with a diameter of around 40 nanometers, is brought into close apposition with the plasma membrane through the action of 3 important proteins that form the so-called snare complex. The snare complex is composed of synaptobrevin, which is the so-called vesicular snare, syntaxin, which is the plasma membrane snare, and SNAP25, that's closely associated to syntaxin. Through binding to each other with very strong strength, they pull the plasma membrane and the vesicle membrane into extremely close apposition with each other. And it may in fact be that there's already a pre-bound where the inner leaflet of the plasma membrane is already in continuum with the outer leaflet of the plasma membrane of the synaptic vesicle membrane. So this would then be the release competent primed vesicle ready to be released. It's brought into close contact with the plasma membrane through the action of the snare complex, and it's waiting for an action potential to come. The action potential invades, opens the voltage-gated calcium channel, calcium floods in, and calcium then binds to the calcium sensor, synaptotagmin, and other vesicle-associated protein. Synaptotagmin has 5 binding sites for calcium, and when calcium binds to it, it changes its conformation quite dramatically. It changes the way it interacts with the snare proteins and perhaps also with the phospholipids in the immediate environment. The action of calcium on synaptotagmin opens the fusion pore, and so there's an opening that's created where the inner leaflet of the synaptic vesicle membrane becomes continuous with the outer leaflet of the plasma membrane, and that creates a pore where the contents of the synaptic vesicle are now in continuum with the extracellular space. There's a diffusion of the vesicle of the neurotransmitter across the cleft, and it can then bind to the postsynaptic receptors, driving postsynaptic potentials. So the core elements in terms of molecular mechanisms of fusion are the priming and docking of the event, setting up the vesicle through the snare protein so that it's release competent, and synaptotagmin then acts as the calcium sensor that converts the action potential and calcium influx into the vesicle fusion event itself. And all of this occurs then on the microsecond timescale. after the neurotransmitter has been released from the synaptic vesicle is empty, and there's no neurotransmitter left in it. Before that synaptic vesicle can be useful again in terms of releasing neurotransmitter, it must then be refilled. And the refilling process of synaptic vesicles occurs through an ATP-dependent process. A so-called vacuolar proton pump, an ATPase, uses ATP to pump protons, that is, charged hydrogen atoms, into the vesicle. It then becomes acidic inside the synaptic vesicle, and it also becomes positively charged. And so, just like there's a membrane potential across the plasma membrane, there's also a vesicular potential across the synaptic vesicle membrane. And it's a combination of the acidification and the electric potential here that gives energy for the vesicular neurotransmitter transporters that then bring neurotransmitter inside the synaptic vesicle. And so if this is a glutamatergic synapse, there would be a vesicular glutamate transporter, V-glut, present on individual synaptic vesicles that then sucks glutamate in and concentrates glutamate inside the synaptic vesicle. When the vesicle is refilled, it then needs to move into these sites where it can be released. It needs to interact with the snare complex. The snare complex needs to bring the membranes into close apposition with each other, and we're then ready for another action potential to invade and release neurotransmitter. There are various ways in which this synaptic vesicle cycle is thought to work. One way is signaled here. This is the so-called kiss and run exocytosis, where there's a brief fusion event, a fusion pore opens, the contents are diffusing out, and the empty vesicle then closes, moves aside a little bit, maybe some nanometers away from the plasma membrane, refuels with neurotransmitter, and then redocks. And that's called kiss and run exocytosis. Another possibility is so-called kiss and stay exocytosis, where after the vesicle has fused, it simply reseals and remains docked and simply replenishes its neurotransmitter content while remaining in a docked conformation. Finally, another possibility is that this fusion pore enlarges and the membrane gradually simply becomes part of the plasma membrane. So the vesicle fuses completely and then becomes part of the plasma membrane, from where it can then be endocytosed in a clathrin-dependent manner and then enter into the vesicle cycle. The fastest forms of exocytosis and recycling are the so-called kiss and run and kiss and stay types of vesicle recycling, which can occur on the 50 to 100 millisecond timescale. And so the whole process here might occur very rapidly and help sustain fast rates of neurotransmitter release if necessary. So we've now seen some of the key steps that are able to sustain neurotransmitter release at these extremely fast rates of 100 microseconds or so between the invasion of the action potential into the bouton and the release of the neurotransmitter. And the key steps are the priming and docking of the vesicle so that it's ready to react in case of calcium concentration increases. So the snare complex forms a key feature in an ATP-dependent process that brings the membranes of the synaptic vesicle and the presynaptic membrane into close apposition with each other. The vesicle then waits there, release ready, until a calcium signal appears that's driven through the activation of voltage-gated calcium channels, driven by the voltage depolarization of the action potential as it invades the bouton. The calcium binds to synaptotagmin, and it's that change in conformation of the synaptotagmin protein that allows the vesicle to fuse with the plasma membrane, releasing the neurotransmitter content. Now, it should be remembered that the exocytosis, the release of a vesicle, is a stochastic process. A given docked and release-ready vesicle may have something like a 10% chance of being released when an action potential invades the nerve terminal. This means that synaptic transmission is unreliable, but it also means that the process can be highly regulated. We can increase the efficacy of the synaptic transmission by, say, increasing the amount of calcium influx into the terminal, or we could also decrease the amount of neurotransmitter release. And it's that ability to regulate synaptic transmission that's thought to be extremely important. And so in the next slides, we'll take a little look at how one might regulate neurotransmitter release. The first and perhaps most obvious way in which we can control neurotransmitter release is by controlling the amount of calcium that enters into the nerve terminal. You'll remember that we have an extremely steep dependence of calcium, where if calcium goes from 1 to 10 micromolar, then we go from release rates of 0.1 to 1,000. So a factor of 10,000 increase in calcium. And typically, during an action potential invading the nerve terminal, we think that we're reaching something like 10 micromolar of calcium. And so by increasing or decreasing the amount of calcium, we can also increase or decrease the amount of neurotransmitter being released. And there's an extremely steep dependence where we go with the fourth power of calcium. So small changes in calcium can give rise to very big changes in the amount of release from the presynaptic terminal. So how do we change the amount of calcium that comes into the presynaptic terminal? Well, there are two obvious ways. We can change the action potential waveform. So if a typical action potential has a duration of something like 500 microseconds, the calcium influx occurs during the repolarization, and by the time the membrane potential is hyperpolarized, the calcium influx has come to an end. So by increasing the duration of the action potential, we can then increase the amount of calcium influx that occurs, and thus we increase the amount of neurotransmitter release. And that can be done by regulating potassium channels. So potassium channels govern the repolarization phase of the action potential, and if we simply reduce the amount of potassium current, then we'll delay the action potential waveform and will increase calcium influx into the presynaptic terminal, and hence increase neurotransmitter release. And that's one way in which neurotransmitter release is really being regulated physiologically. Another obvious way is by acting on the presynaptic calcium channels themselves. Increasing the open probability or increasing the voltage dependence of these calcium channels will of course increase the amount of calcium that floods in to the presynaptic membrane. And that's another way that's used heavily inside the nervous system to regulate neurotransmitter release. Other possibilities in terms of regulating neurotransmitter release are of course how much neurotransmitter is actually inside a synaptic vesicle. That's controlled in part by the proton pumps that give rise to the gradients that drive the uptake of the neurotransmitter into the synaptic vesicle, and so you can change the efficacy of the proton pumping or the efficacy of the neurotransmitter transporters. And both of those are used to some extent to regulate neurotransmitter content inside synaptic vesicles. And although synaptic vesicles typically have a size of something like 40 nanometers, there's also some variance. So there are some bigger vesicles and some smaller vesicles. And so that's another way in which one can control the amount of neurotransmitter that's being released. Presumably, a big vesicle would have more neurotransmitter content than a small vesicle. Another key way in which one can release different amounts of neurotransmitter is by controlling the amount of vesicles that are being released in response to calcium. So the complex machinery associated with the docked synaptic vesicle here and the calcium channel can then change its composition in terms of what proteins are present, what isoforms. There are many different isoforms of these different proteins. So, for example, synaptotagmin has something like 15 different genes that code for it, and they're expressed differentially at different synapses. Equally, there can be phosphorylation events, so that can change the structure of these different proteins, and maybe even the physical arrangement, whether the calcium channels are close to the synaptic vesicles or far away, can make a big difference to whether the calcium is likely to drive a lot of neurotransmitter release or whether the probability will rather be lower. So we can change the release probability and its sensitivity, of course, to calcium, and that's a key way in which neurotransmitter release is regulated physiologically. Equally, of course, it's possible to change the number of vesicles that are present on the presynaptic specialization. So if we only have one vesicle that's there and it has, say, a 10% chance of being released when an action potential invades, we can also put in more synaptic vesicles. We can simply dock more and more synaptic vesicles, put them close to calcium channels, and each one of these might then have a 10% chance of being released in response to an action potential. And clearly, the more vesicles we have that are in a release-ready state, the more likely we are to get neurotransmitter release and large amounts of it when an action potential invades the presynaptic terminal. So we've now seen the key events responsible for coupling an action potential to neurotransmitter release. The depolarization of the bouton causes the activation of voltage-gated calcium channels on the timescale of 100 microseconds or so from the action potential invasion. Calcium rises in the immediate vicinity of the synaptic vesicles that are release-ready and competent. The calcium binds to the synaptotagmin. 5 calcium ions bind to a synaptotagmin molecule, high affinity, and they then drive the fusion of the synaptic vesicle membrane with the presynaptic membrane. The whole process involves some 50 or so proteins, all of which play an important role in synaptic release. And they can be regulated in a large number of interesting ways that are physiologically important. And in the next few lessons, we'll consider in more detail how the neurotransmitter release is regulated by the action of other neurotransmitters acting on the nerve terminal, and also by the recent history of action potential firing in that nerve terminal.